In this session, I'm going to discuss with you how genetic engineering has been used for developing transgenic plants against abiotic stress, mainly salt stress, or in other words, osmotic stress. You know, so when we talk of stress, as you are aware that all the living beings, whether animal or plants or microbes, they require an optimal uh, conditions to live, and anything beyond those optimal conditions, either towards higher or towards lower side, gives stress to the living organism. Now, this is a general observation for all the living beings. Similarly, plants they require an optimum conditions to grow, whether it is temperature, water, or even salinity. If they increase or decrease, this leads to stress. Now, again, all living beings they have their way to meet this challenge. For example, if we look at uh, how how the plants respond to these stress conditions. If the conditions are very harsh and extreme, they become lethal. Okay, lethal means the plant dies. But if the conditions are within the tolerance limits, then the plant survives. We call those levels of stress sublethal levels. Now, although the plant survives, but then it tries to manage, it tries to respond to those stresses at molecular level, that is at the cell level, and those cell level changes respond to the biochemical changes or leads to biochemical changes, and these biochemical changes leads to the response at the cellular level, and when it happens in more than one cell type, then it leads to physiological response, and then whole plant level change. So this sequence of events from molecular to biochemical to cellular to physiological, and then the whole plant, you know, it leads to adaptations, you know, adaptations. And that's how you see different organisms adapting themselves to different conditions to live, and they flourish. Uh, For example, if you look at the desert plants, they have their own mechanisms to survive through the acute conditions of drought and temperature. So they have adapted themselves by bringing about certain changes in them. And as you see here, if the stress level are lethal, then it leads to whole plant senescence or the plant death. You know. So the challenge to the plant scientists is to decipher these changes which are happening in the plants at molecular level. You know. So when we look at the cell, you know, the cell, all cells of a plant, they have this mechanism operating that they perceive the signal of the stress at the cell membrane level. And then this signal, which is perceived here, is transduced to or taken to nucleus, of the cell. And then in the nucleus, the response to that stress signal is generated. So you have a very clear mechanism, which we understand now, is the signal perception, how signals are perceived, and how signals are taken from membrane to nucleus, what we call signal transduction, and then the nuclear response to that signal, you know, uh, which means that the nuclear genome, the nuclear genome induces certain set of genes which helps the plant, or whose product helps the plant to withstand the stress conditions. So this nuclear response leads to the generation of the very specific mRNA in response to this signal, and then they are taken to cytoplasm where the proteins are synthesized. Now these proteins, so this is up to the molecular response where the certain genes are switched on, switched off, or overexpressed or underexpressed in response to the signal. And then these gene products ultimately make proteins, which leads to the biochemical changes or biochemical response, and then which constitutes basically the cellular response. And when this happens in millions of cells of an organ, it constitutes the physiological or the whole plant response. So that's how a plant cell responds. In turn, the plant responds to different signals, which it perceives. Okay. So, a lot of efforts have gone in to decipher this. What is the mechanism of signal perception? What are the genes involved here? How the signals are transduced? And what are the genes which are getting expressed, or whose expression is stopped, or overexpressed or underexpressed, so that if we know how the gene machinery is responding to these signals, then we can think of, uh, developing a technology. Now, when it comes to genetic engineering for abiotic stress tolerance, uh, this is before almost 3 decades ago. This whole work was done when, as I told you, that there was no functional gene available to be used for genetic engineering for any trait. Uh, but then there were few observations. For example, under stress conditions, plants were known to accumulate these kind of metabolites, mannitol, proline, glycine, butane, you know. But then how this happens was not clearly understood, you know. And then, of course, subsequently, there are certain transcription factors and then the proteins which can play multiple roles in more than one stress response pathways. They were worked out, but these discoveries were at a later stage, you know. So I will focus on one aspect which was studied in greater detail, that is the proline. So this is a normal observation that under stress, abiotic stresses like drought, cold, high or low temperatures, and salt stress, or in other words, if a plant has an osmotic stress, you see the accumulation of proline and also mannitol and glycine butane. But we will focus on proline, how this happens, you know. So by mid 90s last century, proline biosynthetic pathway in E. coli was well understood. That is, in E. coli, it involves three genes, Pro-A, Pro-B, and Pro-C. Pro-A is responsible for glutamyl kinase. And so this Pro-A gene, which codes for glutamyl kinase, converts glutamate to gamma glutamyl phosphate. And GSA dehydrogenase, which is coded by Pro-B, converts glutamyl phosphate to glutamate semialdehyde. And then there is a chemical reaction, that is cyclization of this product, which gives to the product known as P5C or pyroline 5 carboxylate, which then acted upon by the product of Pro-C, that is P5CR, pyroline 5 carboxylate reductase, okay, which then converts P5C to proline. Now, this mechanism was well understood in E. coli, but how it happens in plants, that was not understood. So, a group in US, they started focusing to understand or decipher the proline biosynthetic pathway in plants. It's a long story, but then I will briefly mention that what was done is they used the cDNA library from root nodules of mothbean. Now, why that root nodules of mothbean were used? Because these nodules are produced as a consequence of rhizobium plant interaction, which does the nitrogen fixation. And in these nodules, they found high level of proline accumulation. So proline accumulation actually is seen not only for abiotic stresses, but also for biotic stresses. In both the stresses, plants try to overaccumulate proline. So a cDNA library was constructed using mothbean root nodules, and then they were screened using Pro-A as a probe, and Pro-B as a probe, and Pro-C as a probe to find out whether they have plants have homologs to these genes. So when they did this kind of analysis, what they found that the gene which they picked up using Pro-A as a probe, uh, it the gene sequence hybridizes 50% of the gene which they got from plants. And when they use Pro-B as a probe, then this Pro-B hybridizes to the remaining part of the plant gene. So in plants, what was found that these two genes are fused as one. And this gene was named as P5CS, that is pyroline 5 carboxylate synthase. So this gene they characterized functionally and both gene-wise. So it's a bifunctional, and it codes for a bifunctional enzyme. It means this enzyme has glutamyl kinase activity as well as GSA dehydrogenase activity. So over the evolution from E. coli to plants, this is what happened, that these two genes got fused, having both the functions of Pro-A and Pro-B. So what happens in plants, the glutamate is acted upon by P5CS and it converts glutamate to glutamate semialdehyde, gamma glutamate semialdehyde, you know. So, and then they got a gene P5 equivalent to P5CR Pro-C that also was there in plants. So basically, instead of three gene activity in plants, they found it's a two gene activity. So they have a bifunctional enzyme, P5CS and P5CR. Okay. And also, when they studied uh, that this enzyme or gene expresses uh, more under stress conditions. So further, when they looked at the mechanism part of it, they found out that in plants, the proline is synthesized using two pathways. One is the substrate is ornithine. It uses ornithine delta aminotransferase. which converts ornithine to P5C, pyridine 5-carboxylate, and which is converted by P5Cr to proline. This is one pathway. And another pathway is where glutamate is used as a substrate, acted upon by P5CS, and it converts it to P5C. They also found out that the plants have both the pathways which work independently, in the sense that under normal conditions, this is the pathway which is followed to make proline, because proline is one of the important amino acids under normal conditions. But when the plant is under stress, then this pathway is switched on to make more proline. Now, this corresponds with the observation I told you that plants have plants over-accumulate proline under stress conditions. So that is because of the switching on of this pathway by this enzyme. And you see more proline getting accumulated. So this was the pathway which was worked out. But then the question was that which of the pathway, this or this, or which is the gene which is key for the overproduction or accumulation of the proline under stress conditions. For example, whether it is P5CS or P5CR or ornithine delta aminotransferase. Now, all the three genes were tested in transgenic tobacco plants to check that if you overexpress this or this or this, which one leads to conferring tolerance against abiotic stress, like salt stress or osmotic stress. So the conclusion of these studies is that it is the P5CS, which is the key enzyme, which is responsible for overproduction of proline under stress conditions. Okay, and they demonstrated in tobacco that in tobacco, for example, this study dealt with under salt stress, transgenic tobacco overexpressing P5CS gene increased proline accumulation. Okay, as compared to the control, which were not overexpressing this gene. And also, it increased the root biomass and it increased tolerance to salt stress. Okay, so it confirmed that if you overexpress this gene in transgenic plants, it does what is expected from this gene to do. You know, later on, the same thing was repeated in rice. So this gene was overexpressed, and almost similar observations were made here that it led to proline accumulation, increased plant growth, shoot length and root length and weight, fresh weight, shoot and root fresh weight. So, and the increased tolerance to salt stresses. So this was validated in rice as well, but this rice was japonica. As you know, rice has two main varieties. One is indica type and another is the japonica type. So this work was done in the japonica type. Now, the question was that what the proline does to give plants osmotic stress tolerance. So one of the functions which proline does is the osmotic adjustment. And then it also protects intracellular proteins, you know, from degrading, you know. And also, it was found that hydroxylated scavenging, the other roles which proline does, and the protection against UV-induced peroxidation, and the structural and functional integrity of protein as I mentioned. So these were some of the roles which were assigned to proline, that what it may be doing. Also, you know, uh, this was studied from a different angle. That is, if you uh, underexpress this gene, then what happens? So, when in Arabidopsis, this gene was suppressed by using antisense technology. Uh, in these transgenic plants, what was observed is that this suppression leads to defect in inflorescence development, leaves are hypersensitive to osmotic stresses, and morphological abnormalities, retardation in the vascular system development. You know, so it means the proline overproduction or proline as a whole, not only required for osmotic adjustments or protects plant against osmotic stresses, it also regulates developmental processes. Because when you silence this gene or reduce its expression using antisense technology, it leads to these kind of deformities and also making plant hypersensitive to osmotic stresses. So this added another angle to the story, that it means that proline has many functions. Means it also regulates the developmental processes. That after the rice genome sequence was known, so that genome was or Arabidopsis, first the Arabidopsis genome was known. So they tried to look through the genome and they found that there are proline responsive elements in the 5' upstream region of variety of genes, you know, and these responses they call PRE responsive genes, you know, though, and the similar genome analysis was done in other plants as well, and they found that proline also influences the developmental processes through their response elements, you know. So in my lab, we try to do use this technology and form and generate abiotic stress tolerance using this technology, that is P5CSG. Now, why rice? Rice is, as you know, is a staple food, and more than half of the world population depends on rice. So therefore, if we can change this phenotype, then we will be able to protect the plants or protect the productivity of these plants, you know. So in my lab, we did this study, you know. So if you see, this is just to give you an idea that how the rice productivity world over happens. Almost all Asian countries, they are into rice production. This is an Indian scenario. So the point I'm trying to make here is that we'll be focusing on this, that how the indica cultivar, which we used IR50, which is very popular cultivar in South India, and it is grown and eaten there. So the the technique which so these are the genes which were used. One is this is the plasmid, PUBP50S, which has a promoter, and then the coding region and the NOS terminator. And then you have a selectable marker gene in this plasmid, that is PHX4, that is hypermycin phosphotransferase gene. This is the selectable marker, and this is the gene of interest, you know. And the method which was used in this is the biologistic method, gene gun, you know, and what happens in gene gun is this: you coat the particles with these two plasmids uh, together, and then bombard it through using this machine, which we call gene gun, and these are the target cells here. So these particles are shooted onto those, and eventually uh, they enter into the cell, enter the nucleus, and release the DNA. You know, and and then you have this. You know, so once you have bombarded the particles onto the cells, then you should have this protocol from callus to whole plant, shooting, rooting, and then uh, the plants are taken to greenhouse where they flower and show seed set. You know, so these protocols need to work in your hand to generate. Now, what you see here after bombardment, when the calli are put on, when the calli are put on selectable medium with hydromycin, you know, you see the black cells, the zones. These are dead cells, untransformed cells, and these are the one which are originated from transformed cells. Means they are able to survive in the in presence of hydromycin or the antibiotics. So in subsequent cultures, only this part is taken and put on for regeneration, shoot regeneration, then the root regeneration, and then when the plant grow, they are taken to plants are taken to greenhouse. While this is going on, these plants need to be looked at whether the transgene has gone in and integrated into the plant genome. So this is confirmed by doing southern analysis, you know, where you take plants genomic DNA and cut it with the restriction enzyme, which very specifically releases, or within the, which very specifically releases the coding region, you know, and then this DNA is run on gel, transplotted, and then it is probed with uh, the coding region of the, in this case, P5CS. Okay, so what you see here, that this probe is hybridizing to the genomic fragments, okay, indicating that the gene has gone into the DNA and it has been integrated, you know. So these A-line two plants were taken, so it shows single hybridization, and these two lines, C and E, shows two hybridizations. So this indicated that this line has single copy integration, and these two have the two copy integration. So once the gene has been shown to get integrated, you have to see whether it is expressing and making RNA. Uh, so these hydromycin resistant plants, which were selected, RNA was isolated, and cDNA was made, and then the PCR was done using uh, gene-specific primers, and you can see the amplification of the known region in the transgenic plants. And in controls, there was no amplification, and this is the control. Okay. This was also further confirmed by northern analysis. You know, northern analysis is done to check the presence of transcripts into in the genome. You know, so here again the RNA was extracted and run it on the gel, and then they are transported onto the membrane, and then the membrane was then hybridized to the probe, and the probe is the coding region of the P5CS. And you can see here controls, there is no signal at all, and in the transgenic lines you can see uh, 3 and 4, 5 and 6 uh, they are hybridizing and showing the presence of transcript, you know. So it means the transgene has been integrated and it is expressing, it is making the DNA, it is making RNA. Now the next uh, point to be shown was that these RNAs are making protein. So that was checked by doing Western analysis using P5CS specific antibodies. And you can see here, two lines were checked. Uh, and this is control. You don't see any, you know, hybridization with control proteins. And in line A and B, you can see clear hybridization with the uh, uh, protein made by the transgene previously as well. Okay, so what we have shown here that transgene has gone in, got integrated, and expressing, making RNA, mRNAs, and then it is making protein. Now, whether this protein is active and doing its job, it was checked by looking at the proline accumulation. Now, if you see here, under normal conditions when you grow the plants and look at the proline contents, you can see there is difference. This is the control, and then this is line A, and this is line C. And you can see significant high levels of proline uh, made under normal conditions. And when you expose the plants to the salt, higher levels of salt stress, you can see it gets over accumulated here as compared to the controls. Now that is because in these plants you have an endogenous copy of rice P5CS and also an additional copy from what we have pushed in. So both the genes are functional, and therefore you see over accumulation here as compared to controlled plants. So it means the gene has gone in and it is doing its job, what it is supposed to do. Then, then we checked the effect of transgene on growth and development of plants. So if you see here, uh, under normal conditions and under salt stress conditions, the parameters checked was root length, root biomass, and the fresh weight. So, I mean, this data corresponds to this actually. If you see visually, you have the control plant root biomass, and you have root biomass of the transgenic plants, which is a lot more as compared to the normal plants. So, the transgene is affecting expression, transgene expression is affecting the root biomass, making more of roots as compared to the controlled plants, under stress conditions. And this is one of the parameters for abiotic stress or salt stress rates. Under salt stress, the plant's root biomass increases, and that also happens with the with um, um, okay, then if you give salt stress to the plant for 20 days, means you stop watering, or you give higher or irrigate with water with having high concentration of salt. So this is what happens. The control plant dies within 20 days, and the transgenic lines or transgenic plants, it continues to grow, and it gives flowering and seed set. So the seeds were collected here in the T2 generation, T1 generation, and then they were further analyzed, and how it was. So see, the germination process is very, very sensitive to abiotic stresses, So if you see uh, the control plants under normal conditions, they show almost 95% seed germination. But when you give, when you grow the seeds under salt stress, it is blocked 100%. There is no germination, seed germination observed. But when you see the transgenic seeds from transgenic plants under normal conditions and also under stress condition, the seed germination was almost same in both the lines A and C, indicating that the transgene expression is protecting the seeds against the stresses, and the germination processes germination process goes on like now. uh, when the inflorescence or the seed set process was evaluated, again, if you see here, the normal plant or the control plants were put under normal conditions, they give you about 10 to 12 or 11 inflorescences per plant. And when the salt stress is given, the plant dies within 20 days. So there is no question of uh, any seed set or any inflorescence, basically. But if you see the transgenic lines here, the number of inflorescences is less under control condition as well as under stress. As I showed you there, the plants survive and they show inflorescence and the seed set. Both the lines, the same phenomenon is happening, means under normal conditions, there is a reduction in the number of inflorescence. But if you see, the under stress, control plant dies. But here, you see 50 to 60% retention of inflorescence is there. So if you compare from here, the plant is protected against this, and they are able to show fluorescence. And of course, this fluorescence was matches with the seeds per plant. So if you see here, for example, a control plant under normal conditions gives you about 200 seeds per plant. But under stress, the plant dies, so there is no seed here. But transgenic lines, if you see, they give almost 150 to almost 200 seeds, almost same as the control, with a little bit of variation, number of seeds per plant. And if you see here, transgenic lines under stress, though the number of seeds per plant goes down, but if you compare from here, it is almost 50% produce is saved, So, so it means a farmer which normally, if he would have grown this seed stock, would have lost its produce 100%. But if he goes to this seed stock, he can still retain its 50% production, which is a lot. So to that extent, this technology basically has worked, in protecting plants against abiotic stress. Now, another aspect of this transgenic biology is to see that whatever transgene has been pushed in, whether it is following what kind of segregation patterns. As you know, all the genes, they follow Mendelian pattern of inheritance, like 3:1. So we try to screen out the plants. So it was a very complicated study, done, wherein these many number of plants were screened for presence of the gene, FICS gene, and by dot blot southern analysis. It means the DNA was isolated from these many number of plants, and they were blotted onto membrane and then probed, and That is what was observed, that 73 out of 132 were PFAS positive, 74, 49, 64, 38. So if you see 3 lines we checked, 2 lines followed a 3:1 ratio. So the transgene which got integrated is following Mendelian pattern of inheritance. You know. Now, another question, which is associated with the transgene integration, what we want, that this gene should not enter in a functional gene. If that happens, it is going to mutate that gene, and it can be counterproductive in the sense that you may introduce a trait, but then at the expense of the gene where the transgene is getting integrated in between a functional gene. So therefore, these kind of plants need to be studied uh, for the determination of transgene integration, where it has gone. So that is being determined by plasmid rescue approach. So what is this plasmid rescue approach? Is we have sent the plasmid which gets nicked in between and become linear and then get integrated into the genome. So what is done? We try to rescue this plasmid out by cutting it by an enzyme. We know what is the single restriction site in the plasmid, and the other site will be on this side of the genome, and the other side will be here. So you will see genomic DNA when restricted within restriction enzyme, which is cutting the plasmid in one. For example, you have if you cut it with say BMH1, so or BMH1, and the one side it will cut here, and another will be in the genome, and similarly here one in the genome, and you ligate it and then transform into E. coli, and then try to see the cells which can grow on ampicillin. Now, a portion of the plasmid which has the origin of replication and the ampicillin resistant gene will be able to amplify, and such cells were then amplified and the DNA was isolated, and then it was checked for like this. So you have a scenario wherein you have the part of this UB promoter intron in the coding region, and then it enters into the genome sequence, So this gets amplified. So now you can remove this from here to here, and then this fragment was subcloned, So where it has part of PFAS gene, and this is the genomic DNA, So Hindi3-01 fragment was taken out from here, and then it was sequenced. So the idea here is that if you know this sequence and then try to match it in the genome sequence, that what it means, where the gene has gone, then you will know that whether your transgene has gone into a functional gene or non-functional gene or some repetitive region. So when the sequencing was done and it was matched with the genome sequence of the rice, we have, the indica rice, the japonica rice, and this is the query sequence, and the green zone, what you see is the deletions actually, but rest is one-to-one matching with with both indica as well as the japonica rice. And this is the region actually, which is referred as the hotspot region in the genome. Hotspot region means where a lot of deletion and additions keep on happening. This region doesn't code for any gene actually. So this was also, in a way, good conclusion that the gene has gone into a region where there is no functional gene, and it is not going to affect any other thing than the phenotype which one we are referring to. So to conclude this part, basically we could transform this local variety from India uh, by this gene from Vigna contrifolia, as I told you, moth being gene, using ballistic method, and the transgenes were analyzed, and the site of integration was determined into this place, you know, and uh, and this country is not assigned any region. Okay, so this is the kind of success story, very early success story, and the first of its kind, um, where um, a gene was discovered, which was able to change the phenotype of the plants. And subsequently, this gene was isolated from other systems, from not being almost every plant has this gene and shows the same proline biosynthetic pathway in all the plants. Only thing is if you overproduce and if you add additional copies of the gene to the to any crop plants, you can make it to withstand or tolerate uh, higher levels of stresses as compared with the control. So this technology has been used, and if not in India, at least genetically modified crops have been generated in US and other countries. So thank you very much. With this, I end this session also.